Good afternoon, everybody, and welcome. My name is Jane Langford. On behalf of the Nordics and Baltics Nutricia team, I'm really pleased today to chair this session, focusing on the role of symbiotics in immune modulation and allergy management. We're thrilled to be joined by so many of you and healthcare professionals from 7 countries around the local region. So I'm really excited today to be joined by 2 of our experts from the research team at Nutricia Research in Utrecht, the Netherlands. I'd like to introduce Dr. Magdalena Timerson, who is our senior team leader, and Professor Leon Knippels, our science director of allergy. And they will present on the role of symbiotics in immune modulation and allergy management. So before we start the presentations, we'd really like to take a moment to gauge your current perception on the role of symbiotics in allergy management using this first poll. So the question we're asking here is, are you familiar with the evidence to support symbiotics in allergy management? And we have 3 answers here, which you can choose. Firstly, no, not at all. I'm not aware of symbiotics, nor the role they may play in the clinical management of allergies. Secondly, I have some awareness, but I still need to be convinced about the clinical relevance. And thirdly, yes, I'm familiar. I know what symbiotics are, and I actually already use symbiotics in my allergy patients. So we'll give you now a few moments to think about that and answer the questions, and then it'll be really interesting to see the results that we get. Great. Okay, so we see the poll results here. So I think this is really interesting. So 23%, so nearly a quarter of the people who've joined us today aren't familiar with the clinical evidence to support symbiotics in allergy management. The majority, almost 2/3 of you, have some awareness, but still need to be convinced. And 1 in 5 of you are familiar and currently using symbiotics in your allergy patients. So this is great. So this really shows us that we've got a bit of a job to do here. And I'm very confident that throughout the presentation, we'll be able to answer a lot of these questions and hopefully encourage you to use symbiotics in the future. So we'd like to make a start, firstly, by introducing you to a short video and welcome to Nutricia Research. And then I'll hand over to Magdalena in Utrecht to start her presentation. Thank you and enjoy.
Good afternoon, very welcome to all of you. And actually we are presented to you from the R&I Center in Utrecht on the 4th floor. So as you've just seen in the video, that's the top floor of this fantastic building in which we have different laboratory and also office space. Today we would like to introduce you to the field of the role of symbiotics in immune modulation and allergy management. So first of all, some background knowledge on the immune system. As you may all be aware, but possibly not, the immune system is all about balance. So we really need to have our immune system to keep us healthy. And how does the immune system do it? Basically by accepting all the healthy cells in our body. Another important task of our immune system is actually to tolerate all kinds of different triggers that we do encounter during our day. So basically, from breathing the air to the food that we eat, we all need to tolerate it because we need the food to grow. We need the air, obviously, to get oxygen. But we also need our immune system to fight off intruders, pathogenic intruders, but also to fight off sick cells in our body. So as you can probably appreciate, this is a very delicate balance and very difficult for the immune system to keep this balance. Unfortunately, sometimes this balance is tipped, which may result, for example, if the tolerance is failed, it may result in allergy. You can have allergies towards food, but of course also to environmental triggers like grass pollen or other inhalant allergens. For example, if the immune system fails in accepting the healthy cells of the body, this may result in autoimmunity. I'm sure you're all aware of diseases like rheumatoid arthritis, in which the immune system is actually fighting against their own body. And if the fighting against the pathogenic intruders and sick cells fails, this results in infections, but also in cancer. So basically, tumor cells are the sick cells which should normally be eliminated by our immune system. If this isn't done properly, you will get sick. So hopefully you can appreciate that the balance of your immune system is really, really important at all times. So from the early days of life till later on. So today we would really like to focus on allergy. And as you know, the severity of an allergy condition can vary over time and also in severity itself. So what do we have? We have, for allergy management, we basically have extensively hydrolyzed formulas and amino acid formulas for the allergic infants. And what I would like to focus on today is actually the bottom part of this slide. So we will introduce you to the field of the prebiotics, probiotics, and then together the symbiotics. And we'll take you through this step by step. So we envision this to have a dual approach of active allergy management. So first of all, you need a controlled exposure to the protein. So basically the immune system doesn't need to see the allergen, in this example, cow's milk. But our second part is actually to generate the right environmental conditions. So you would like the immune system to see the allergen, but by setting the environment using pre and probiotics, we can steer the immune system. As I just told you, it's all about balance. So an immune system can react in many, many different ways. And if you provide an allergen combined with certain environmental combinations like pre or probiotics, you can actually manipulate the balance of your immune system, which is something we would like to achieve. So, and why is the nutrition during early life so important? Well, when you're born, your immune system is there, but it really, really needs to develop. So it's very early stage. And for development, obviously, you need triggers. So you need exposures, for example, to cow's milk, but also to other proteins. And as you can appreciate, all the infants will encounter bacteria, but also viruses. So also pathogenic triggers will be there. So this all combined with the environmental triggers, the food antigens, will actually shape the immune system of the infant. And those early days are important by providing the triggers and setting that balance. Because if you tip off the balance early in life, this may result in diseases later in life. We know that the shifting in the balance may result in things like allergy, but also onset of diabetes later in life. So nutrition can actually influence this and train the immune system. And how would it do that? So basically, as I already mentioned, so the diet will actually help in training the immune system, because that is actually the triggers that the immune system will recognize, or so it will learn whether it needs to react, for example, towards a grass pollen or shouldn't. And not just the grass pollen, also other proteins and other triggers. So a few things that are important, which I already alluded to a little bit, is, for example, the differences in the diet, but also the exposure to pollutants. So it really matters if an infant is actually growing up in an industrial area, in a city, or on a farm, where the microbial exposure is really, really different, and your exposure to pollutants is very different. Next to that, we know that the mode of delivery, whether it's a cesarean section or a vaginal delivery, is also very important in shaping the immune system of the infant. Another thing we know really influences the development of the immune system is antibiotic usage. Obviously, it's needed because sometimes you do need the antibiotics to really fight off the infection. However, it will also shape your immune system towards possibly a direction you do not want to go. So luckily with nutrition, you are able to influence a little bit this training of the immune system. So the first, a little bit too impatient. So the first nutrition an infant is exposed to, obviously, is human milk. We know that human milk is actually the best for an infant to grow. And it's very important in preventing all kinds of illnesses. And as I mentioned earlier today, obviously, we will focus on allergy and asthma. And we know that breastfed infants are protected for a large part for allergies and asthma, but also of all kinds of other diseases, as is depicted on this slide. So there's many publications on that as well. So what is actually in human milk? So many years, the laboratory here and also other scientists all over the world have studied extensively the components that are in human milk. Because if we know that, obviously nature is our best teacher. So if human milk is actually so good for an infant to grow, because that's the nutrition it will get, we should learn from that how we can help infants that are not able to be breastfed or later in life. So what is in there? So from the top to the bottom, what you can see, there are actually cells in human milk. Cells like lymphocytes, macrophages, and granulocytes, which are actually all part of the immune system. And those cells are actually able to protect against infections. Next to the cells, there's also proteins obviously in there, things like antibodies, growth factors, and cytokines. And as you probably all know, that antibodies from the mother that are given to the child will actually really help in protecting the infant. Because the immune system is still very immature of the infant, the maternal antibodies will definitely help during the first weeks and months of life. And then the purple parts, where we can show bacteria, are actually also present in human milk. We've seen that bifidobacteria and lactobacilli are actually there, and they are important for colonization of the infant's gut and also important in shaping the immune system. Next to the bacteria, there are also oligosaccharides present in human milk. We call these actually HMOs, which stands for human milk oligosaccharides. And there are hundreds and hundreds, more than 200 different oligosaccharides actually present in human milk. And they have very important functions also for colonization, shaping of the immune system, but also by affecting the gut barrier function itself. Next to those things, we also find fatty acids in there. So the saturated fatty acids, the polyunsaturated fatty acids, those are also important for your membrane structure and maturation of the immune system. Obviously, things as protein, carbohydrates like lactose, and also minerals and vitamins are present in human milk. So it's actually obviously that unique combination of human milk that is shaping the immune system, but also helps in colonization of the infant's gut early in life. So what else is important to know? So I already mentioned a few times the colonization of the gut. So why is the gut important? Obviously, that's where nutrition enters your body. But it's also good to realize that at least 70 to 80% of your immune system are also located in the gut. And not just the immune cells are there, so also your gut microbiota are there. And there are like 100 trillion bacteria actually present there. Next to that, there's also a lot of neurons there. So approximately 100 million neurons are also located in that same organ. And it has a surface of approximately 30 square meters. So you can imagine it's a huge organ which has a lot of influence, not just on the immune system, but also of the rest of the body. So this might seem like a very complicated figure. It's much more abstract than the previous one. So I will talk you through it. So this is actually the gut microbiota and the immune system in a situation where tolerance exists. So if you go from left to right, what you can see here, what we've depicted, are the human infant type bifidobacteria. So those blue ones, those are the bifidobacteria, the good guys. Those bacteria will actually metabolize non-digestible oligosaccharides. By doing that, they will secrete the short-chain fatty acids, for example, lactate, butyrate, and acetate. These short-chain fatty acids will actually have an influence on the pH in your gut. And normally it's between 5 and 6, as indicated here, which will then actually reduce the growth of pathobionts, thereby providing protection. But these short-chain fatty acids actually have many, many different functions. So will also directly stimulate cells of the immune system, which are depicted here and called DCs, dendritic cells. Those dendritic cells are actually reaching through the lining of your gut lumen. So this is your epithelial layer depicted here of all epithelial cells, and your immune cells can actually reach through and sample directly from your lumen. And that's how the short-chain fatty acids can directly influence immune cells. Then as a subsequent result, those dendritic cells can travel to lymph nodes and then have all kinds of effects on T cells, which will have an influence on the balance of Th1 and Th2. So again, that immune system balance between Th1, Th2, allergy, non-allergy. Next to that, short-chain fatty acids also stimulate the epithelial cells directly. So not only via the dendritic cells, but also on the epithelial layer, it can have an effect. And then epithelial cells can also have direct effects on dendritic cells. So it's all actually connected. And it can also act via an indirect way, so by feeding other beneficial bacteria. So not necessarily the bifidobacteria, but other beneficial bacteria, which may in turn also influence the epithelial cells and the immune cells directly. So as you can hopefully appreciate from this slightly complicated picture, that everything is connected and everything will influence each other. There are direct influences of the bacteria, the bacterial products, but also indirect via other bacteria or via metabolites. And it all actually comes down to a balance in Th1, Th2 responses and regulatory T cells in your immune system in the gut. So if we now focus a little bit on the development of the gut microbiota, obviously it starts when the infant is born. And as you can see here in this picture, it really matures over time. So similar as to the immune system that really needs to mature and to develop over time, the gut microbiota will also mature and develop. As you can see, the diversity increases with age. So within the first 12 months, it really expands from only a few bacteria to many different strains. And very early in life, you can see it's really colonized, we generally say, by the bifidobacteria. So that's the normal picture. And there has been a lot of research developing the microbiota from allergic versus non-allergic infants, for example. And this is an example where you can see the microbiota of a healthy infant versus a cow's milk allergic infant. And we call this the healthy microbiota versus the dysbiosis one. And as you can see, there is actually a shift in which bacteria are present. So this is correlated to the cow's milk allergy. So in a dysbiosis, we talk about the decrease of the size and distribution of the microbacteria. So this is actually what then happens in an allergic setting. As I just said, so the dysbiosis is actually marked by a decrease in certain bacteria, so the beneficial ones. That's actually depicted here on the left. So in the previous one, you could see a lot of blue ones. So there's a lot of good bifidobacteria. However, in an allergic setting, there's much less bifidobacteria, which then results in a decreased secretion of your short-chain fatty acids, which actually will lead to a change in pH. Thereby, the environment will be different and pathobionts actually have more room to grow. The environment is more beneficial for them to grow. As you can also see, the epithelial layer is not as healthy as it was in the healthy state. You can see there's actually, we call this the disrupted barrier function or leaky gut. What you can see is there's really more spaces in between the cells, which actually means that pathobionts are more easy to travel through the layers. And also all the other interaction that we discussed in the other graph, you can see there's effects there. So there's direct other effects directly on your immune cells, which lead to a different T cell balance, which may lead to the differences that we see later on. For example, the IgE that we always see in allergy. So what can we do here So basically, what we would like to show you today is the pre and probiotics that we have investigated for many, many years now. So first of all, a prebiotic is actually a substrate which is selectively utilized by host microorganisms which are conferring a health benefit. And to be classified as a prebiotic, there are certain criteria that you need to fulfill. So first of all, the prebiotic needs to survive the acidic conditions of the stomach, because otherwise it will not have the effect on the microbiota further down. It also needs to evade the digestion in the small intestine. So it needs to be able to travel through the stomach and the small intestine, because basically it needs to be fermented in the colon by beneficial bacteria, like the bifidobacteria or the lactobacilli. And of course, the prebiotic should improve host health, because that's the definition of a prebiotic. So which prebiotics then can we use in early life? So as we already alluded a little bit, we are very focused on human milk, what's in there. And as I already said, there's many, many different structures in human milk, many different oligosaccharides. And as you can see here, there are short chain human milk oligosaccharides and long chain human milk oligosaccharides. And basically what we use in our laboratory is the short chain GOS, long chain FOS, and a 9:1 ratio. And this is all inspired on the composition as found in human milk. And hopefully you can appreciate that it's not just one form and the other, but it's really a combination of many, many different oligo structures. So there are actually two prebiotic mixtures that we will use, and they will also come back later on in the presentation. So the one on the left is the short chain galacto-oligosaccharide and the long chain fructo-oligosaccharide. And on the right side, you can see the picture of the fructo-oligosaccharide. The reason for that is that galacto-oligosaccharide is actually derived from lactose. So for the very severe allergic infants and the amino acid formulas, we only use the FOS 9:1 ratio, basically because of safety profile. And then, of course, we need to know how many prebiotics should you actually use. So this is a study already from 2002, showing that these prebiotics that we would like to use actually promote the growth of the beneficial bacteria. But you can see in the graphs on the left one, you see the percentage of the amount of the bifidos, and here on the lactobacilli. The dark blue banner is actually the reference of the breastfed infants. And then there are three groups. One group is actually getting formula without the oligosaccharides, so 0 grams per 100 ml. And then there's a group receiving the 0.4 grams per 100 ml. And you can already see that there's a difference, and there's more bifidobacteria present in the features of these infants. And then if we increase the concentration to 0.8 gram per 100 ml, you can actually see that the bifido is coming into the breastfed range. And slightly comparable for the lactobacillus, as you can see. So also the 0.4 and 0.8 was actually also enhancing the bacteria towards more in the breastfed infant range. So this was just the bacteria that were actually present in the features of these infants. Obviously, we're very interested in the clinical outcomes. So these are results showing the atopic dermatitis of infants either fed with the GOS-FOS formula versus control. And what you can see on the left, so the purple bar is actually the incidence of atopic dermatitis after 6 months, and the green bar is the infants receiving the GOS-FOS, so the prebiotic formula. And you can see that the incidence of the atopic dermatitis is indeed reduced. What's very interesting is that even after 24 months, so long after the intervention has stopped, there is still a difference in the incidence of atopic dermatitis. So still the infants that are receiving the formula with the prebiotics, the percentage of those infants having atopic dermatitis is really much, much less compared to the control group. So that was the part on the prebiotics. Of course, there's also probiotics. So what is the definition of a probiotic? A probiotic is actually a live microorganism, which then, when it's administered in adequate amounts, will confer also a health benefit on the host. Obviously, there are many different bacteria that could be investigated as a probiotic strain. So the results I'm showing you here is also all from preclinical work. So this is an allergic asthma model in mice. And what you can see here is actually the number of cells in a bronchoalveolar lavage. So that's basically mice that have become allergic. And then you will look at the composition of the fluid in the lungs. And we depict here the different immunological cell types. So lymphocytes, neutrophils, macrophages, and eosinophils. And what you can see is that normally the Shem sensitized mice, so that is basically the negative control, those mice don't have any immune cells in their lung fluid. If you make a mouse allergic, that's the second bar, you can see clearly all the cells there. And then if you treat these mice with different probiotics, you can see there's different effects. So there's actually 2 probiotics that do show a reduction in the immune cells in your lung fluid. Next to that, we also investigated the ear swelling and the serum over IgE. And what you can see is that, again, the BREVE M16V, which is indicated by the red arrow, is the one that is able to actually reduce the allergic symptoms. So not just by diminishing the amount of immune cells in your lavage, but also by diminishing the ear swelling and reducing the level of IgE. So this would be a very good candidate to actually look into for the clinic. And that's what has been done over the past years. So a little bit more details on this Bifidobacterium breve M16V. So this bacterial strain is actually isolated also from infants. And it was also the most commonly found bifidobacteria found in human milk. So this would actually really also add to the safety aspect of this probiotic. So it was selected among 400 different strains, and since then it has been used in clinical study, even including preterms, since 1994. It has also been studied in various allergic populations. So basically, this probiotic strain has a very well-recorded safety record in all these studies. And it also has demonstrated in neonates, so in all kind of different target populations as well. So how do we combine this now? So we envision the symbiotic concept. So what we would like to achieve is the best of both worlds. So combining the prebiotic and the probiotic. So by doing this, a symbiotic is actually a mixture of specific pro and prebiotic that will actually, so the prebiotic will improve the survival and the implementation of specific live microbial dietary supplements. And so it really needs to be complement to each other. We'll show you that in the next slide. So what you can see here, so why is 1 + 1 actually more than 2? So on the left side, you can see you can choose a prebiotic and a probiotic, which will both have beneficial effects. However, obviously what we would like to achieve is a synergistic effect. So we actually would like to choose a prebiotic that will actually stimulate the growth and the activity of the probiotic that you have chosen, thereby enhancing the probiotic effect as well. So that's actually what we've done. So we are combining the BREVE M16V with the GOS-FOS 9:1 ratio. Again, starting with our preclinical work, what you can see here again is mouse models, where you can hopefully appreciate that again, we're looking at ear swelling. So it's a measure of immunological response. So this is our control. So actually the allergic response. If you provide the mice just with the BREVE, it is reduced. If you provided with a prebiotic mixture, like the GOS-FOS, it is also reduced. However, if you then combine this pro and prebiotic, so we call this the symbiotic engine, this reduction is even better. So this basically indicates that there is a synergistic effect between our prebiotic mixture, the GOS-FOS, and our probiotic M16V strain that we have picked. Next to the effects on those, we also looked at the effects directly on the immune cells. As I've explained in the cartoon where we talk about the tolerance versus the allergy, all the metabolites and the bacteria can also have direct effects on, for example, the dendritic cells. So the dendritic cells are actually very close to the epithelial cells and can actually sample the gut lumen, are also exposed to the pre and probiotic mixture. What you can see here is that dendritic cells normally can produce different cytokines. IL-10, for example, is a tolerance-like cytokine. So if you produce that, you would counteract allergy. And dendritic cells actually exposed to either BREVE on its own, which are the open circles, or to the BREVE and the GOS-FOS together, so the symbiotic mixture. You can see that there is a little bit of IL-10 production if you expose the cells to the probiotic itself. However, if you expose the cells to the combination of the pre and probiotics, you can see a huge increase in your IL-10 production, which would actually mean a direct effect of the symbiotic mixture on the immune cells in the gut, which would then favor an anti-allergic immunological reaction. So now some more clinical data. So this is actually the SYNBOT study, which includes 90 infants with a score at more than 15. They received the extensively hydrolyzed formula, and they received the formula for 12 weeks. And then we have a group without the symbiotic and a group with the symbiotic mixture. And obviously, we will look at the severity of the atopic dermatitis. But of course, we also always investigate the intestinal microbiota composition. So first of all, the differences in the scorerat. So what we show here is the results after 4 weeks, 8 weeks, and 12 weeks. And I need to stress that this is specifically in the IgE-mediated atopic dermatitis. So this is specifically in the IgE positive population, these results. The purple bars are the control infants, and the green bars are the infants receiving the symbiotic extensively hydrolyzed formula. As we show the delta scorerat, you can see there is an increase in your symbiotic mixture, which would actually indicate a positive effect of your symbiotic formula. And next to the scorerat, we also looked at the bifidobacteria. Here on the left side, you can see the week 0, week 1, and week 12. And as you can see, the blue bars are the control infants. There, the percentage of bifidobacteria is actually declining over time, whilst in the infants group receiving the symbiotic formula, you maintain the same level of bifidobacteria. And on the right side, we've depicted the diaper dermatitis. And as you can see, the symbiotic group receiving the, so the symbiotic group is actually experiencing less diaper dermatitis compared to the placebo group. So that's basically 60 versus 86%. Interestingly, if we then look 1 year later, so this the intervention was the nutritional intervention was 12 weeks. So 1 year later, these same infants were actually investigated for the use of asthma medication. And we also recorded the asthma-like symptoms, things like noisy breathing and frequent wheezing. And again, as you may see from these graphs, that the infants receiving the symbiotic formula actually experience less noisy breathing and less frequent wheezing. So again, indicating a positive effect of the symbiotic mixture. Here on the right side, you can actually see the change in asthma medication in the symbiotic group versus the control group. The green part is the increase, that there's an increase in asthma medication. And hopefully you can appreciate that the increase in the symbiotic group, there are some infants indeed where it's increasing, but that percentage is much less compared to the infants receiving the control formula. So this is a little bit more detail from the same study. So again, the 1-year follow-up data, again, more in numbers, the reduction of the asthma medication. So the two groups, the groups receiving the symbiotic formula versus the placebo formula. And as you can see, for example, here, the asthma medication is really reduced. So from 33% in the control group to only 13.9% in the group receiving the symbiotics. So to conclude on this part, basically the extensively hydrolyzed formula with the symbiotics has clinically proven to be safe. It's also relieving some allergy symptoms, and it actually meets the criteria set by the various guidelines. So the Gosfos BREVE is safe, it will support the adequate growth, and it also restores a more breastfed-like gut microbiota pattern. On top of that, it has a beneficial impact on atopic dermatitis infants, and it actually may prevent some asthma-like symptoms in infants and reduce asthma medication. So the last part of this presentation, I would like to focus on amino acid formula with symbiotics. So first of all, the ASSIGN study, which included non-IgE cow's milk allergic infants. So the intervention this time was 8 weeks instead of 12 weeks in the previous study. Again, we will look at the different fecal composition, so what kind of bacteria are present, looking at stool characteristics, gut health markers, and also, of course, looking at clinical symptoms. So first of all, the bacteria results, as you can see here, so start of the intervention and later on, but you can see here the gray box is actually the healthy breastfed reference. What you can see is the start is roughly similar between the two groups, and then the group that receives the amino acid formula with the symbiotics is more comparable to the microbiota of the breastfed reference group compared to the group not receiving the symbiotics. And the same goes for the Adobacteria. So here the rectile and the cholecystitis. What you can see is that indeed, again, the group that receives the amino acid formula with symbiotic is more similar to the breastfed group compared to the group receiving the control formula. So if we then focus to the adverse events, what you can see here is the basically any adverse events and also the infections and ear infections in particular. You see here on the left side, this is the group receiving the neocate SYNEO. So basically the group receiving the symbiotics, there were no ear infections reported, whilst there were 7 reported in the control group. And also for other adverse events, we focused on the dermatologicals, but you can see here there is a reduced usage of dermatologicals in the group receiving the symbiotic mixture compared to the control group. Then there's one last study I would like to share with you today. That is the amino acid formula with symbiotics in the PRESTO study. So these are infants actually with IgE-mediated cow's milk allergy compared to the non-IgE-mediated cow's milk allergy in the previous study. This intervention was much longer. It's actually 12 months, as you can see here. And we've looked here at the tolerance to cow's milk and also at the immune gut microbiota markers. So first of all, what you can see here is there's no clear differences in the outgrowth of the allergy, you can see that there is a tolerance development towards cow's milk in all infants after 12 months and also after 24 months. So the symbiotic containing formula is basically comparable to the other formula. So there is still tolerance development towards cow's milk. And as you can see, the gut microbiota is again more comparable to a breastfed group in the infants receiving the symbiotic mixture compared to the group receiving the control microbiota. So next to the microbiota, we also looked at the infections occurring in these infants. And here we can see again, as we've seen in the previous study, especially for ear infections, here we look at the general infections. There is a reduction in number of infections in the group receiving the symbiotics compared to the group receiving the control formula. And this is actually alluding to that a little bit more again, so that basically if you provide the infants with formula with symbiotics, that you do see a reduction in infection and also in antibiotic usage. So indicating that the symbiotic mixture is actually beneficial for your immune system. So in summary, if we now look at the studies that have been performed with the extensively hydrolyzed formula and the amino acid formula with the symbiotics, we can conclude that it's clinically proven to be safe in these allergic infants. It does promote appropriate growth in the infants. It does meet all the guidelines. We do see some symptom resolution in the cow's milk allergic children, especially also the atopic dermatitis. We see a rebalancing of the microbiota. So it's really balancing much towards the breastfed microbiota. There's definitely positive effects on atopic dermatitis, infections, and also medication use. And not just the dermatologicals, but also on the antibiotics usage. So our vision on active allergy management is that we really need to educate the immune system of very young infants. As mentioned in a very, very early slide, it's all about balance. So of course, there's tolerance and there's allergy, and you would like to restore a balance if it's tipped off by whatever reason. So for that, what you do need is some protein exposure. So we do need the allergens to be present. But most of all, we need also to modulate the gut microbiome and to stimulate direct and indirectly via modulatory components, the development of the immune system. So for that, I hope I have showed you today some results on our specific prebiotic mixtures and probiotic, and especially the symbiotic mixture of our pre and probiotic mixture together to restore this balance. So with that, my last slide, I would like to thank you for attention and give the word back to Jane.
Thank you, Magdalena. So I would really like to thank Magdalena a lot for such an excellent and informative presentation. It really did summarize the reasons for choosing the particular pre and probiotic blends we had, and really the evidence that we have both preclinically and clinically to support these ingredients in the allergy management and immune modulation. So before we move to the Q&A, we'd really like to gauge how the presentation went. And in terms of thinking about the similar question to the first poll question, let's see if we've moved anybody's opinions further. So what we'd like to ask you now is, do you believe that symbiotics can improve health outcomes in cow's milk allergic infants based on what you've heard today? So firstly, no, I'm not convinced. Secondly, yes, I think there may be evidence to support that we can improve health outcomes, but it's strain specific or in certain amounts. Or yet, thirdly, yes, all the data is relevant and really does have a clear clinical benefit. So we'll give you a few seconds to answer these questions. Great. Well, that is convincing. So I think in the first poll we had nearly a quarter of the audience were not aware of the data to support symbiotics or the evidence in cow's milk allergy. whereas now only 1% of the audience, and we have nearly 150 participants here, so I think that's great. Well done, Magdalene. I think it's clear from this is that the audience considers it strain specific. So maybe not all probiotics and prebiotics can play a role in the benefits in allergy management. So I think we'll move on to the panel discussion now, and really it'd be great to pick Magdalene and Leon's brains in terms of some of the questions which we received prior to the webinar, and we'll wait and see which questions do come in also from the audience. So as mentioned previously, please do, if you have a question, type it into the Q&A box. We will be monitoring this, and we'd be really interested to hear comments or questions about what's been shared over the last 40 minutes. So firstly, I'd like to ask one question. You've summarized really nicely the data that we have for the particular strains which we use within our Nutricia formulations. But I'd be really interested to know, why did we choose the Bifidobacteria over some of the well-known lactobacillus probiotic strains?
Yes, I think that's a very good question, Jane. So there are several reasons. So one of the reasons is the preclinical data that we have. So obviously we compared many different probiotic strains in the mouse allergic asthma model. Indeed, it's not just the M16V that's performing really well there. There's also other probiotic strains. However, if we then look at the human situation, since the M16V is a strain that is naturally occurring in the infant, and this strain is also found in human breast milk, this is actually one of our preferences then to go with that probiotic strain.
Perfect. Thank you so much for that. And how much of the strains do we think is most beneficial for infants to receive? Is there an amount that we have proof that we see reduction of allergic symptoms and the other benefits?
Yeah, that is a very good question. So the initial studies looking at the different concentrations, so the very first ones were obviously the prebiotics, where we did different concentrations. For the probiotics, because I was thinking the other way around, because this is really for the probiotic question, right? Not necessarily for the
sorry if I wasn't clear.
Yeah,
yes, on
no, you were. I was actually
So I think there are two aspects to this question about the numbers. One, of course, we did also preclinical work, which we did. We tested several dosages of the Bifidobacterium BREVE, and that resulted also in a certain number at the end in our products. But if you look, for instance, especially at human breast milk, if you look at the numbers of Bifidobacterium BREVE, but also other strains present in human milk, this is very comparable with the amounts of the numbers of live bacteria that we have in the products that we make. So the numbers are really related also to the physiological, let's say, aspect of the numbers that are present in human milk, and also based on preclinical data. And normally we get to 10 to the 9th by heart in our products. So that's the background why we chose these numbers.
Perfect.
Hopefully that helps.
Thank you. No, it does, because I think we do get questions about how much of the formula, do we see a benefit? Because obviously different ages of children take different levels of the different formulations. So we want to know that we're actually giving something that benefits over a wide range. So no, that's really interesting. Yeah. And can we talk a little bit? I think Magdalene did a lovely overview of the evidence that we have. Do we have further data on longer term follow-up of children who've received these symbiotic formulations in early life?
Yes, so you basically are alluding to the long-term effects. Yeah, I think we have some nice data on that. So not just on the atopic dermatitis, but also on the asthma-like symptoms. So I think that as a scientist, I find it really remarkable that if you have an intervention of only like 8 or 12 weeks, obviously it's very early life. So I think that's why it's such a significant period of time. If you have your intervention then, that it really have effects later on the asthma medication and also on the asthma symptoms or wheezing-like symptoms. So that is definitely seen, and also for the atopic dermatitis.
Yeah, and it's quite fascinating, isn't it, that maybe even taking a formulation for only 6 or 8 weeks early in life can see some effects later. So I think there's a lot more work, obviously, that you and the teams in Utrecht and around the world are doing to understand the mechanisms of action of these different ingredients.
Exactly, and I think it really pinpoints that the critical, the timing is so critical, and that early life is so important, even for 1, 2, 5 or 6. So that is also why we have our clinical studies also have follow-ups, definitely after 5 years or 6 years. Because when children are a little bit older, it's also easier to establish whether a child suffers from asthma, yes or no. Obviously, with an infant, it's much harder to tell. So yeah, I think it's fascinating how we can influence the immune system in such early life and then has such beneficial effects later on.
Absolutely. So to the audience, I guess, watch this space. There will be more coming from these trials. We are following them up for a number of years. So I'd like to move to a couple of the questions that we have in the chat. So thank you to the audience for asking these. So a question from Louise, is there a difference in the probiotics in human milk based on a mother's ethnicity? So do we see differences in bacterial populations depending on ethnicity or where in the world they are?
That is a very good question and very interesting. As for the probiotic or for the bacteria present, I don't know from the top of my head. What I do know is that indeed human milk definitely differs, and I'm sure a geographic region will definitely influence as well, because of course it's also influenced by the diet and the microbial pressure that the mother encounters. So it's definitely influenced by that as well. One thing we also know from human milk is that it's not just the environment and the diet of the mother, but also whether a mother experiences infections, as you know, that will also really influence the composition of the human milk. So excellent question. And yeah, we're still investigating that as well. And also over time, because human milk, obviously you have your first periods and then later, and there's also differences there.
Yeah, and I think,
can I maybe add something? Oh, and you need to mute again. Sorry. Well, yeah, because totally true, Martha, what you were saying. But maybe also to add, because I assume that there will be differences, as you explained, but also based on the fact that we're now getting more and more insights in the composition of human milk. We know we have so-called secretors and non-secretor mothers. So some mothers do secrete and give, let's say, certain human milk oligosaccharides to their infants, whilst others don't. And that's just based on genetic background. That's the difference. We have 4 types of different in human milk. So probably there's also difference potentially in the bacteria that these mothers give to their infants. Mm-hmm. But I think also, especially because we don't know exactly how these bacteria enter up in human milk yet, from a science point of view. So it's very difficult to explain why they are there. So I think the explanation that Martha gave for the difference in pressure, microbial pressure, diet of the mother can influence the bacterial status. But we do know there are differences in human milk. That's known.
And I think that goes to one of the other questions we have in the Q&As about the types of prebiotic oligosaccharides as well. So how do they vary over the first year of life as a mother's milk adapts? It's similar to the bacteria. I guess the prebiotic and the human oligosaccharides do change also. Yeah.
Yes, they do.
So it's quite a complicated business, and I think obviously as a company specializing in breast milk substitutes, trying to understand this, we will never be able to be as complicated as the wonders of human milk. Which
exactly.
Is why this is the gold standard.
I think nature works fabulously, as always, and human milk is just the best nutrition example that we have. And as I said in the introduction, there are so many different oligosaccharides actually present, but also in different ratios. So some are more there, other less, but there could be differences also in binding of those oligosaccharides, of effectiveness of those oligosaccharides between different categories. So we're definitely not there yet. Many things to discover.
No, exactly. And I think it brings to another question. There's a lot of interest in these human milk oligosaccharides and possibly the use of synthetic human milk oligosaccharides, which are now being used in different formulations. So why do you think, what's the difference between symbiotics compared to these HMOs, or how do we compare in terms of efficacy and potential benefits?
Yeah, so I think first of all, that's what I always learn from Leon, is that not every HMO is a prebiotic. And as I try to explain also in the seminar, it's really the combination between the pre and probiotic that makes the symbiotic. And it's not that every prebiotic mixture can just be the best substrate for your probiotic. So that is actually a balance. Yeah, and then as said, there are so many different human milk oligosaccharides, and some of them we just don't really know yet exactly what they do. Then, of course, there's also those different mixtures, different combinations. So, yeah, there's definitely still a lot to investigate there.
Yeah, I think you're totally right, Marcel. But maybe also to add, I think also if you look at the whole discussion about human milk oligosaccharides, of course, they are important. That's clear, because that's why they are there. And one of the most important ones that's very often used, of course, is 2FL as an example. But we also know that some women, as I explained before, you have secretors and non-secretors. Some of them don't even secrete 2FL. So it's not that all women have 2FL in their human milk. And I think that's important, and it's important to know. So from a biological point of view, for some women, it's important to have 2FL, other ones don't. And we don't know why. That's one thing. The other thing is that, as Marcel explained, we have all these structures. There's so many structures of human milk oligosaccharides present in human milk. That's why we try to mimic it as well a little bit with this Gauss-Foss mixture. So we have the long chain and the short chain structures, but it's not one long chain and one short chain. It's a whole bunch of different short chain and long chain structures, and try to mimic as much as possible differences in human milk. Although it's, of course, it's not human milk oligosaccharides.
Yeah.
But that's also how we approach, let's say, the complexity of the composition of the human milk. Maybe to add more, sorry.
Yeah. No, thank you. Thank you. I think it's really shown it is more complicated than one or two ingredients. There's so much going on in there that, and as the science evolves, we're understanding different things every year on this. So it's definitely going to be an evolving concept. And I think it brings me maybe to the last question, because I'm aware of the time and everybody probably wants to go and start their evening before too long. But when do you think, or do you think the guidelines will change in the coming years to reflect the evidence for symbiotics in allergy management?
Yes, that is a complicated one. I know they're working on that. The European Society is working on that. I think one of the difficulties luckily, I'm not the one who made the guidelines. Let's say that first. I think one of the difficulties is all the differences in the study. So as I tried to explain in the seminar, sometimes you have IgE-mediated allergy versus non-IgE-mediated allergy. So the study populations can be different, what is measured. And I can imagine that it's going to be very complicated to have like one general guideline for this. But there are a lot of studies at least indicating towards beneficial effects towards, for example, reduction in eczema, atopic dermatitis. So yeah, there's definitely some good news, and I'm sure Leon wants to add something.
Yeah, you already said it. First of all, we are not policymakers. We can provide as scientists, let's say, the evidence for other people to make a policy. So I'm not going to burn myself to say anything about potential policies. But I assume what Marcel said is totally true. Not every probiotic is the same. So you have strain specificity. You have also specificity with the prebiotics, also with symbiotics. There's more and more evidence also for other strains that they have an influence on the immune system, on allergy reduction, but to put it overall in very strict guidelines, that's not so easy. But already the WHO a couple of years ago at least recommended that you could use prebiotics. And although it's not, the evidence is not so strong, it's difficult to compile it all on one plate, let's put it that way. So you need very specifically to look at which probiotic, which symbiotic, which studies, which population, and that's a difficult task. But from a science point of view, I trust that prebiotics and probiotics, if sufficiently studied, do have a very good effect and do have a preventive effect. And also in light of, let's say, more and more knowledge we have on the disturbed microbiome, how you can modulate the microbiome, that it has a positive effect on not only on immune development, but also on the development of the gut in general. I think these are all aspects that will be taken into account, but we need big clinical studies. And that's, of course, the bottleneck of for a lot of policymakers, that we need more studies.
No, but, I think it's a very good way to end there, Leon, because clearly there's a lot more to do. I think we've come a long way, and I think from the data that's been shared today by Magdalena, really shown clearly the safety profile as well of these ingredients. I think now we have such a long history of use that, you know, we're reassured that they do have very, very excellent safety profiles and are bringing additional clinical benefits, but we do need to be clear that these are sometimes not the primary outcomes of studies, so more research is always required, and I think that will always be the conclusion of scientists around the world. So I'd really like to wrap up there. Thank you both so much for your time today and for really so clearly sharing the data on this state of the art in terms of the symbiotic engines. I'd like to thank all of the healthcare professionals who've joined us out of their busy days. I know everybody's incredibly busy, so we appreciate you taking the time. We will be sending an evaluation form shortly, so we'd love to hear how you felt the presentation went and whether this was valuable for you. And the webinar recording will be available very soon, and that will be available either as a link directly to you as you previously registered, or available on the local Nutricia site.